specific surface chemistries. He'll be talking to us today about chemically defined synthetic surfaces for stem cell expansion. Please make Laurence welcome.
Thank you. And thanks to Robert for inviting me to speak today. I thought I would go a little bit more general than just speak specifically about hematopoietic stem cells. So I'm going to describe for you some fairly simple systems that we've applied predominantly to mesenchymal stem cells, and we've done a little bit of work with embryonic stem cells. So I'll just run through that quickly. Just introduce our why we're interested in this area and what our drivers are. At least from our perspective, are a base coating, and in our case these were always polymeric coatings, so synthetic, which has low non-specific interactions with biomolecules. We want to use highly specific synthetic signalling molecules for cell attachment, spreading, expansion and differentiation. We want to be able to covalently immobilize both the base coating and the signalling molecules. We want them to be applicable, the coating method to be applicable to the sort of materials that people are using to expand cells. Most of those are polystyrene. Sterilizable using conventional methods like gamma sterilization and a long storage life at ambient temperature. So there's some of the requirements. And what I'm going to describe to you today is essentially a simple free radical grafting process. It's a polymerization, compatible with tissue culture where scalable manufacture. We can use a wide range of monomers and a wide range of polymeric surfaces and a wide range of culture formats. And these are the sort of culture formats we've been working with on a fairly small scale at the moment, but as I said, the technology is scalable to larger formats. These are microcarriers, for example, and obviously tissue culture flasks and multi-well plates. I'm not going to go too much into the chemistry of how we do this, but basically we take a tissue culture polystyrene or any other polymer surface. I'm going to describe first a method for making surfaces that have low protein absorption and cell attachment, and this is, if you like, the base coating that reduces non-specific interactions. One of the monomers we use is a monomer called polyethylene glycol methacrylate. We do a homopolymerization to generate a graft polymer coating which we can sterilize with gamma irradiation. And we've done that on all those four formats that I described to you earlier. It's essentially a neutral surface, no charge groups, and has a high density polymer brush coating, if you like. Just to continue on the theme of low cell, low protein attachment coatings, there's a number of instances where you want to use that type of coating for in vitro assays where you don't want you want low protein absorption, culture of non-adherent cells, formation of cell aggregates like embroid bodies, neurospheres, hepatospheres, culture of cells that might be activated by adhesion, and cells that might differentiate by adhesion as well. So there's a couple of advantages. You don't need a different plate depending on whether you want a low protein or low cell attachment. Simple method of fabrication, gamma sterilizable. So here we have a comparison of protein absorption. In this case it's human serum albumin on tissue culture polystyrene on a polystyrene surface on a commercially available low cell attachment plate and on our surfaces. And you can see that we get a very low HSA absorption, showing that the non-specific interactions of the base coating are very low. We also get a very low cell attachment on our coatings. Here's thermohydrocell, which is commercially available. Corning ultra low attachment. Here's one of our surfaces. And comparison of HeLa cell attachment on TCPS and another was commercially available NUNC surface. These surfaces are also low attaching to sticky cells like MSCs and also embryonic stem cells. These particular cells are taken from an embryonic stem cell pieces colony that's been treated with noggin for 14 days. And you take a Pisces colony, put it in a low attachment plate, and it forms a neurosphere, which isn't attached to the coating at all. At this stage, if we like, then we do a simple activation step, very widely used chemistry, EDC and NHS activation. We immobilise, we get an NHS surface, we immobilise the peptide on overnight, and then we get an RGD essentially containing surface, which we can also gamma sterilise at that point. When you look at, for example, how cells attach, and this is just some examples for fibroblasts. This is the concentration that we use to do the immobilisation. This is the cell attachment compared to TCPS. So at very low surface concentrations of RGD, you get very low cell attachment. As the surface concentration increases, you get much higher cell attachment, in this case with fibroblasts, higher than you get with TCPS. We can quantify the amount of ligand that we have there on the surface. We use a europium assay. We actually label the peptide and then quantify how much is on the surface. So you can see that if we start from this concentration of peptide in solution, we end up with in the order of 6 nanomoles of peptide on the surface. And that correlates identically with the cell attachment. If you look at the way the cells look on the surfaces, tissue culture polystyrene, as you increase the concentration, you start out with these cell aggregates, which are basically there's no interaction between the cells. This is a study that's almost finished. So we're taking our RGD surfaces, we're using commercially available bone marrow derived mesenchymal stem cells. We're expanding those in various media, and we're using 3 in this case, 2 chemically defined serum replacement medias here, and a serum-based standard growth media for MSCs. We're comparing expansion rates and retention of differentiation capacity through the 3 lineages, as you've seen earlier, doing multiple passages in 3 media plus differentiation, our synthetic surface versus TCPS, and looking at markers. This is at the end of 7 days expansion in the 3 media, mesenchymal therapy and the growth media, so you can see that cell morphology is very similar, irrespective of whether it's TCPS or the synthetic surface. These are the 2 chemically defined media. This is the serum containing media. When you look at the growth rates as a fold expansion, you can see that the mesenchymal media gives the highest fold expansion rates, and what should also be obvious is that, again, we see this 60% reduction in the fold expansion of the cells. We haven't fully analysed the markers to know whether the markers are retained preferentially on the synthetic surfaces, but that data will be coming soon. The other 2 media gave reduced fold expansions, and here you can see the serum containing growth media. So at least with these cells, the chemically defined media actually gave a much better outcome than the serum containing media on both TCPS and synthetic surfaces. When you look at the differentiation capacity, if you like, this is on the synthetic surface. There's the cell morphology. The cells will differentiate after expansion down the adipogenic, osteogenic and chondrogenic lineages, and the result that we get is very similar depending whether it's the synthetic surface or on TCPS here. So very similar results for both of those cases. Because we're interested in expanding cells at volume, we've also been doing quite a lot of work with microcarrier particles for stirred cell culture. And here's an example with and without serum. These are mesenchymal stem cells at 23 hours. This is the negative control, so these surfaces don't have the cell attachment motif on them, the RGD peptide. These surfaces do, and you can see that cells attach and spread in very similar fashion to the way they do in TCPS, on TCPS surfaces, with the difference being that in the absence of serum, so this is just a basal media that doesn't doesn't have a serum replacement in it, you get lower cell attachment in all cases. We've also applied the same sort of surface to embryonic stem cell derived neural progenitors. So this is a neurosphere, as I showed you earlier, that basically grows in a low attachment plate, forms in a low attachment plate. If you increase the amount of peptide that you have on the surface, the cells start attaching onto the surface in a dose-dependent fashion, and they start spreading out away from the initial attaching neurosphere. And when you add an appropriate growth factor, in this case basic FGF, the cells start proliferating out and differentiating down the glial lineage. A platform surface coating methodology that can be applied to cell culture wear. Hopefully we've demonstrated that we can apply that to a variety of stem cell types. Now this is one signal, and it's clearly useful in a wide range of stem cells. Obviously there are other signals that can be used, and that's one of the ways that we're expanding the surfaces to make them better and define, maintain phenotype. We're looking to basically commercialise the approach that we've developed for low attachment surfaces. We've also done a fair bit of work on thermoresponsive surfaces. So these are the types of surfaces that when you change the temperature, the polymer layer changes its structure and the cells detach either as single cells or as cell sheets. I didn't have time to go through that today. And surfaces for the attachment of bioactive factors. Basically, we like to, as I said, commercialise this, and we like to commercialise particular MSC applications, probably in the first instance. And we're applying this approach, as David has said, for the expansion of hematopoietic stem cells that maintain the HSC phenotype. And I'd like to thank you and just acknowledge funding through the CRC program, the Australian Stem Cell Centre, the Victorian Government, internal funding from CSIRO Biomedical Materials team, some collaborators, Andrew Laslo, George Sow were responsible for the HESC experiments, and collaborators at Melbourne Uni, Mirella Dottori and Elise Pibe were responsible for the progenitor work. And thank you.
Questions for Laurence? Sure.
Can you tell me what is the minimal number of the peptide molecules required for your adhesion polymer surface?
So that's entirely dependent on the cell type. But in this particular instance, we have a total number of 4 to 6 nanomoles. Now they're dispersed right throughout the coating. That's not the concentration that's actually interacting with the cells, because this peptide can diffuse into the coating and attach anywhere along the polymer chain. It's really only those that are directly interacting with the cells that are responsible for the attachment, and that's obviously going to be a lower number.
Have you done the titration? You found what's the minimum concentration required to put in this?
Well, that's the data I showed you earlier. At least with fibroblasts, we've
the mesenchymal cells, the stem cell one.
We're doing that right now. Currently, the mesenchymal stem cells are growing. This is going to take too long. They're growing. We use a concentration that gives us about 4 nanomoles for the MSCs. But different cell types have different integrin expression, obviously. Some cells don't express, the alpha V integrins as highly as other integrins, and so the cell attachment you get would be completely dependent